So today we will talk about phagemids. Phagemids are basically a hybrid between a plasmid and M13 bacteriophage vectors. Although M13 vectors are very useful for the production of single-stranded version of them, but they do suffer from one disadvantage, that there is a limit to the size of the DNA, insert DNA or the passenger DNA that can be cloned within a M13 vector with 1500 base pair, generally looked on as the limit or the maximum capacity, right? So researchers wanted a more complex or a more sophisticated vector for carrying more base pairs, right, of the insert DNA. To get around this problem, a number of hybrid vectors called phagemids have been developed, combining a part of the M13 vector and the plasmid DNA, right? So we can say Phagemid is nothing more than but a plasmid DNA with a part of M13 genome or M13 vector. So first we will talk about an example. PEMBL8, PEMBL8. I call it as PEMBL8. Now, what we did in constructing this, we transferred 1.3 kilo base pair fragment of an M13 vector DNA into PUC8. PUC8 is a plasmid vector, modified plasmid vector, and this piece of M13 DNA contains a single sequence, a signal sequence. I'm sorry, a signal sequence which is recognized by the enzymes. Some enzymes that convert the normal double-stranded M13 molecule into single-stranded before the secretion of new phage particles. Right. So, what do you understand by this? Because this signal, why is this signal so important? Because the signal is still functional even though detached from the rest of the M13 genome. So PEMBL8 molecule are also converted into single-stranded DNA and secreted as defective phage particles. Now all that necessary is that the E. coli cells used as host for a PEMBL8 cloning experiment are subsequently infected with the normal M13 to act as a helper phage, providing the necessary replicative enzymes and phage code proteins. PEMBL8 being derived from PUC8 has the polylinker cloning sites. We know that what are polylinker sites? Cloning sites. Polylinker is the oligonucleotide, short oligonucleotide sequence on the DNA, and within this lacks Z enzyme. So the recombinant plaques can be identified in a standard way, in a standard way on the agar containing XCal. Right. With PML8, single-stranded versions of cloned DNA fragments up to 10 kilo base pair in length can be obtained, greatly extending the range of the M13 cloning system. Right. What we did is simply that we inserted a polylinker. So we know that PML derived from them PUC8, and PUC8 already had the polylinker cloning sites within the LAC-Z gene. So the recombinant plaques, that so we can easily identify those recombinant plaques in a standard way. We can see here version of PML8 is given. Uh, as I told you, PML8 made of PUC8 and M13 DNA fragment. Here we can see PUC8 had an ampicillin resistant gene part and a LAC-Z gene, as we remember, right? And that M13 DNA fragment is there. Its size is PML8 size is length is about 3.9 kilo base pairs. So we will see about the conversion of this PML into the single stranded DNA. in this we saw that a double stranded PM lead was there, and an M13 replication protein comes. An M13 replication protein comes and binds to that M13 region of this PM lead, right? And what it does here is this M13 replication protein replicates the PM lead into a single stranded DNA, right? You can see here, right? It is replicating the P embellied into a single stranded DNA. And when the single stranded P embellied DNA molecules are formed, they are converted into the particles, phage particles, right? Here, P embellied phage particles are formed. So this is the conversion. Yeah, simple, right? Now, it is a very wide applicability, right? In various processes, like in recombinant DNA technology. Various processes are there in recombinant DNA technology, RDT, right? Like in probe generation, phagemids are used in probe generation for it as detectors, as we know, after the hybridization process, when we want to identify those specific sequences that we desire or want, right? And it is also helps in DNA sequencing, mutagenesis study, and phage display systems, right? This phage display systems will be further studied. DNA sequencing, you already know. If you want to know a particular sequence of a DNA, we can use phagemids, right? Mutagenesis study, basically it is studied on mute SGM, right? SGM, that is the site directed mutagenesis, not random. Site directed mutagenesis, we know that we can alter loss or gain of some function of that particular sequence by generating a mutation on that particular sequence, and Phagemid vector has a applicability in that area too, right? Phagemid has certain advantages, like the carrying capacity of phagemids is very great, very high than the phage vectors in comparison to the phage vectors. It has higher efficiency in transformation than phage vectors. They are genetically more stable than phage vectors. So here we are type of comparing them to phage vectors, right? Recombinant phage vectors, right? They are exploited to generate single stranded DNA template for sequencing purposes, right? I just told you they have a wide applicability in the RDT. This one there. They can generate a single stranded DNA template for the sequence. Now, single stranded phagemid vectors inside the phage can be targeted for site directed mutagenesis. I just told you that they, when this phagemid vectors are inside the phage, they target this particular sequence of the DNA and generate some response or the action or the mutation that we desired, right? Or maybe loss of that function or particular alternation of that function for our use, right? Single stranded vectors can be used to generate hybridization probes for mRNA or complementary DNA, right? Probe generation, we just talk about it. Now we will talk a summary of this Phagemid. Phagemid vectors are plasmid having a small segment of a filamentous phage M13. It can be FD or F1 phage, right? Capable to carry up to 10 kilobase pair of passenger DNA or the insert DNA. And we saw example P emballate or the P emballate series of the plasmids and P blue script family plasmid. There are many examples of phagemids. Phagemids generally encode no or one kind of code protein. It has only a gene responsible for only one kind of code protein, or it may not even code for that. Right? P emballate discovered in 1983 are distinguished by the presence of it has a BLA gene, BLA gene, which has ampicillin resistance as a selective marker. It has an alpha peptide coding short segment of beta galactosidase, laxi gene we talked about in last segment, containing multiple cloning sites, right? So intergenic sequence region of phage 1. It also has intergenic sequence region of the phage F1, right? These Phagemid vectors have been used effectively for DNA sequencing with the Sanger dideoxy method, which use the chain termination method. Right? It can perform some function of the M13 derivatives. M13 derivatives like M13, MP1, MP2, 7, 8, MP8, 7 and 8. There are many derivatives, and the advantage, the biggest advantage, I think, that they are smaller than M13 vectors, which is which is which is used for the purification of the DNA, which makes the purification of the DNA very simple, right? Just like in plasmid they have PBR 322. It had a very small size, which helped in purification of it very easy, and it can take very long inserts, and those long inserts have a higher stability in PM plasmid than M13 vectors. And within bacteriophages such as M13, replication is complex. In this vectors, Phagemid vectors like PM8 and P blue script families, they have replications quite simple. Thank you.